Okay, guys, let's see what we can do with this chapter on blood. Let's get into this. So, cardiovascular system in your outline first consists of the following. We have the blood. Number 2, you have the heart. And number 3, we have blood vessels. And those blood vessels consist of the following. As they leave the heart and come back, they would consist of number 1, arteries. And next would be arterioles, A-R-T-E-R-I-O-L-E-S. Then capillaries, and as we come back to the heart, we would have venules, V-E-N-U-L-E-S, and veins, finally. So blood is actually a connective tissue. If you remember when we did tissues, we talked about that. It consists of a liquid portion that we refer to as plasma, all right, and the which is actually the matrix, and the cellular portion consists of various cells and cell fragments. So I'm going to move myself out of the picture here. Okay. Number 3, we have interstitial fluid, or the tissue fluid. Essentially the fluid that you're going to find in between the cells of the body. All right. Blood, number 4, carries oxygen and nutrients and exchanges those molecules at the tissue level, right, at a cellular level, for carbon dioxide and waste molecules that are going to be released back into the interstitial fluid. So if you want to study blood and become an expert on that, then you would be studying hematology. All right? So let's talk about some of the functions of blood. Blood is a liquid connective tissue. 3 major functions being performed here. Number 1 is going to be transportation. We're going to transport oxygen, CO2, nutrients, heat, waste, hormones, and various things like that. Number 2, we have regulation of pH, also body temperature, and the water content of cells. Number 3, blood provides protection against loss of itself, or blood loss, by way of clotting. Also protects against foreign microbes and toxins by phagocytosis from the white blood cells and some other proteins. So some of the physical characteristics of blood, and you can see the picture up on the screen right now. Okay, blood is essentially has the following characteristics. All right, it's denser and more viscous than water. By viscosity, we're talking about thickness. So there's an old saying, something about blood being thicker than water, which is very true. Temperature, 38 degrees Celsius, which is normal body temperature. You don't have to know that number for me, but I do want you to know number 3. The pH of blood normally ranges between 7.35 and 7.45, which, if you remember from your chemistry, is slightly alkaline or slightly basic. Oxygenated blood appears bright red in color. Deoxygenated blood is not blue, but it's a very dark red, almost a purple color. Right? It makes up about, number 5 here, 8% of total body weight. So remember that number, good number to remember. And you should also know here, number 6, that the average volume of blood is 5 to 6 liters in an adult male and 4 to 5 liters in an adult female. So components of blood, as you see on the picture here. First of all, number 1, we have blood plasma, or just what we refer to as plasma. So up on our screen right here, you can actually see here's where the plasma is. All right, plasma makes up about 55% of the volume of blood. That's letter B in your outline. All right, it's a letter A, a straw-colored liquid. So it actually appears a little bit kind of a yellowish or off-white type color to it. Okay. Makes up the plasma itself, letter C, is approximately 91.5% water. The other 8.5% would be solutes. Right? Those solutes are going to include mainly plasma proteins, and you see some other things listed in your outline there, which you're not going to have to know as far as being able to restate anything to me test-wise. Okay. We want to go into the formed elements and talk about the formed elements now. So the formed elements are going to include the following things. There's a couple of tables in your book here. So if I go to this, we can see some of the things here, the formed elements. So first of all, we're going to have erythrocytes, red blood cells right here. So these are the erythrocytes, or the red blood cells. Let me increase the size of this just a tiny little bit. Let's scroll down here. All right. So erythrocytes are red blood cells. Next up, letter B would be leukocytes, or white blood cells. And then we're going to get into the specific types. So if I move to this, we can see some of these. All right, so here's our spellings for these as well. All right, so your erythrocytes here, red blood cells. Okay, leukocytes or white blood cells are going to be listed here. So these are leukocytes, LEU. Right. So we're going to split them up. So our leukocytes or white blood cells, first of all, we're going to have what are known as granular leukocytes, because they contain granules in the cytoplasm. And the following types would include here. So I'm going to go back to this picture where we can kind of scroll through them here. All right. First of all, we have neutrophils right here. Next up, we have eosinophils, which is this one down here. We're going to come back and talk about these later. And finally, we have basophils right here. Okay, you know what, let's go ahead and while we're at it, let's put these percentages in here, and they're going to vary from textbook to textbook. So if you go back up to neutrophils, they're going to make up 60 to 70% of all of the white blood cells. So they are the most common. Add that to your notes. Eosinophils, only 2 to 4% of all white blood cells, and the basophils anywhere from 1/2 to 1% of the white blood cells, which makes them the least common type or the rarest type. The next form of leukocytes or white blood cells are the agranular leukocytes. These do not have granules in the cytoplasm. All right, first one are going to be lymphocytes, which are right here. So the lymphocytes make up 20 to 25% of white blood cells. And it will include some subdivisions, which we'll mention later. T cells with a capital T, B cells with a capital B, and finally natural killer cells, which is already listed in your outline right there for you. And the last type of agranular leukocyte right here would be monocytes, comprising anywhere from 3 to 8%. And you don't have to know these percentage numbers for me. Just know that neutrophils are the most common, basophils would be the least common. All right, the monocytes make up 3 to 8%. And finally, the other formed element we have is right here, and that would be the platelets, okay, which are fragmented cells, which we'll talk about at the end of this chapter. Okay. So if we go back to our test tube here, you'll see that it says the total percentage of volume in blood occupied by the red blood cells right here, and that's referred to as the hematocrit, Hematocrit, hematocrit. T-O-C-R-I-T, hematocrit. All right, so we have plasma, 55%, red blood cells, or the hematocrit, 45%. So off to the side, write this down because we did not include this, but it's in the picture, so make sure you know this picture. All right, we have an area, so 45 and 55 gives us 100%. But there's an area in between those known as the buffy coat. So the buffy coat makes up less than 1% of the total volume of blood. And what's in the buffy coat is very important, is your white blood cells, all those leukocytes we just discussed, and the platelets are all found in the buffy coat. So make sure this picture is one that you have down. I believe I have a picture of it in the outline, so you should be well aware of that. So let's talk about the formation of blood and how it's actually formed. Blood cells are formed by hematopoiesis, right, or also known as hemapoiesis. So just change hemapoiesis to hematopoiesis, which occurs only right after birth in red bone marrow, primarily red bone marrow. And then number 2 says we have several hormones that are going to be responsible for differentiating these cells into the different types that they are. These are called hemopoietic growth factors. And those growth factors would include, number 1, erythropoietin. Just write down that is formed or released by the kidneys. We'll mention it later. Number 2, thrombopoietin, released by the liver and the kidneys. And cytokines, which are just essentially proteins, and we're not going to get into discussing too much about the cytokines. Right? You may cover some of that when you get into physio. So let's specifically just focus right now on the red blood cells, and you can see pictures of them here. There's a little picture where we can actually see the red blood cells and kind of what they look like compared to the white blood cells and the platelets. If we go to this picture here, we're going to see when we look at a blood smear, which is what you'll be looking at, normally would be looking at in lab time, but we'll have to look at slides of these a little bit later on, all right, for lab purposes. This is what a blood smear would look like normally. So if we look at what a red blood cell would kind of look like on its own, first, number one, the erythrocytes are red blood cells. Greater than 99% of all the formed elements, all right, in blood are erythrocytes, red blood cells, RBCs. Okay? There's numbers there for how many erythrocytes you should have per microliter of blood for males and females. You don't have to know that. Obviously, it's higher in males because they have more blood volume. Right? Number 3 states that each red blood cell is a flexible biconcave disc that lacks a nucleus. So when you look at it from the side view, it's kind of like a donut that has the middle of the hole still kind of not punched all the way through it. And the purpose for this is so that they can fold up on themselves and actually go through these smaller blood vessels, the really small ones, which are the capillaries. All right. Cytosol of each blood cell, red blood cell, is going to contain anywhere from in the range of 280 million dissolved hemoglobin molecules. So here's your hemoglobin molecules listed right there, and here's kind of the picture of what a hemoglobin molecule looks like. Each one of them, number 5, consists of 4 globin chains, and these are the globin chains, which are polypeptides, all right, and 4 heme groups. And the heme groups here, all right, are where you're going to find the iron part of blood. So right in the center here, you can see the heme group, which is where the iron is established. Number 6 says hemoglobin can bind reversibly, right, to oxygen to form oxyhemoglobin. And what that means that the erythrocytes or red blood cells can take oxygen, grab it from the lungs, take it to tissues in our body, and drop it off. Right? At the same time, after it does that, number 7 says hemoglobin can also bind reversibly to carbon dioxide to form carbaminohemoglobin. What that does, the erythrocytes grab onto the carbon dioxide after it drops off the oxygen and takes that carbon dioxide to our lungs, where we expel it by exhaling. Right? Hemoglobin also is involved with regulating blood flow and blood pressure. Okay? Now, these red blood cells don't have a long lifespan. They only live approximately 120 days because of the fact that they don't have organelles. And what that does is when these things fold up upon themselves and go through these really narrow blood vessels, the capillaries, all right, they get damaged as they're squeezed through there, and because they don't have a nucleus and the other organelles, they really cannot replace or repair themselves, so they only have a lifespan, once again, of about 120 days. Okay. So, a couple of situations here we have to talk about. Number 10, erythropoiesis, right? So erythrocytes, again, red blood cells. Erythropoiesis is the formation of red blood cells. It begins in the red bone marrow and goes through a couple of different stages of differentiation. I won't ask you to know these. So it starts as a pro-erythroblast. Remember, blast is an immature cell. It becomes a reticulocyte without a nucleus. It enters the bloodstream and then becomes a full-blown erythrocyte. Number 11, hypoxia, which is a condition. If you want to draw a little teeter-totter next to that, you could. Hypoxia, which is a decrease in oxygen to the bloodstream, stimulates the kidneys to release erythropoietin, which is a hormone, which in turn stimulates erythropoiesis. So if we don't have enough oxygen in our bloodstream, erythropoietin is going to be released from the kidneys, which is going to stimulate the formation of more red blood cells. Right? So if you draw a little teeter-totter next to that and write down a couple of things here, first of all. So if we don't have enough red blood cells, or a decrease in red blood cell volume is what's referred to as anemia. And there's several forms of anemia. We won't get into all the different types. We're not going to ask about those, but there's everything from sickle cell anemia to pernicious anemia to iron deficiency anemia, which is very common in females due to their menstrual cycle and the fact that they lose blood on a regular basis, monthly basis. The opposite of that, if you want to add to that, all right, is polycythemia, which is an increased number of red blood cells, which in turn would tend to make the blood a little bit thicker. And polycythemia you will see in people who train at high altitudes and things like that, like Olympic athletes and such. And what happens is they train at high altitudes where there's not enough oxygen, then their body essentially develops more red blood cells. And if they do that and were to, say for instance, train at a high altitude and then draw their own blood and save their own blood and transfuse their own blood back into them before an event such as a marathon or something, when they came back down to, like, normal altitudes like sea level, like we live out here, essentially that's referred to as blood doping. That's the purest form of blood doping, which is obviously against all the rules in the as far as the Olympic committees and other athletic events goes. Okay, so increased number of red blood cells is polycythemia. A decreased level of red blood cells is anemia. Number 12 talks about the two major blood group systems, each one based on the presence or absence of genetically determined cell surface antigens, all right, which are called isoantigens or also agglutinogens, all right? So I'm going to tell you about the two groups, and I'm going to try hopefully to get to where we can see this on the board because I can't put it on the screen here and mark on my screen. So if I bring my camera back up, my webcam with my picture on it, hopefully we can actually see that. So let me see if I can kind of bring that back up here as I pause this first, and we'll see if we can get to the board. Okay, so I'm going to see if I can mark this up on the board so you guys can fill this out on your chart there, and hopefully you'll be able to see this and my big head will get out of the way. If not, I'll move afterward where you can see this. So going up to the board here, we want to fill out the following structures here. So under the different blood types here, under type A blood, okay, which hopefully you can see the screen, okay, the antigen present would be the type A antigen, okay? So a person with type A blood would have the anti-B antibody present, right? So that if somebody with type A blood was transfused with type B blood, that blood would essentially clump up in their bloodstream, and the term for that is agglutinate or agglutinate. Agglutination. And once that blood all clumped up, it would kind of form a dam and stop the flow of blood, which can be life-threatening here. Okay, we'll hold off on the can-receive category for a little bit. If we go to the type B blood, they would have, a person with type B blood has type B antigens. They would have the anti-A antibody. Okay, a person with type AB blood has both antigens. Therefore, they have neither antibody present. Okay, maybe they would have neither antibody. All right, and a person with type O blood would have neither the type A or the type B antigen, so they would have both antibodies present. Okay, and antibodies are what our bloodstream forms when we're exposed to something to fight off various infections and things like that down the road. So looking at this chart, we have to determine what a person can receive blood-wise. So if we're type A blood, we can receive type A, obviously, because we have the type A antigen, but we cannot receive type B because we have the B antibody, all right? This one essentially has both antigens in it, so it has neither antibody. So if we have type A blood, we can receive type A, all right? But because this has the B antigen in it, all right, we can't receive AB. So the only other one we can receive, because if there's neither antibody here, antigen, we can receive type O. All right. So a person with type B blood can receive type B as well as type O. Okay. A person with type AB blood, okay, can receive A. They can receive B. They can receive AB. And they can receive O, all right? Because they have neither the anti-A or the anti-B antibody, so they could receive all forms. So people with type AB blood are known as the universal recipient, right? Universal recipient. A person with type O blood has neither antigen, so they have both the anti-A and the anti-B antibody. So they cannot receive type A, cannot receive type B, can't receive AB, So the only type they can receive is type O. All right? But if you look on the screen, you'll see up here above, under can receive, type O is listed in each of these. So a person with type O blood is referred to as the universal donor. So universal donor would be type O. And then we have another blood grouping system, which I can briefly just describe to you up here. It's called the RH system. Okay? And a person who is RH positive, okay, has the RH antibody present. Okay? All right? Or sorry, the RH, let's change that real quickly, has the RH antigen. All right? So RH antigen, got to fix that. Kind of hard when you're doing it up on the screen to keep checking the screen. Okay? So a person who we say is RH negative does not have the RH antigen. Okay? So they have RH antibody in their bloodstream. Right? RH negative would have an RH antibody once they have been exposed to RH positive blood. Right? So where this becomes a situation is in your outlines discusses hemolytic disease of the newborn. So if a female is RH negative, right, and she is exposed to RH positive blood one time, her bloodstream will form the RH antibody, right? So that if she is exposed a second time to RH positive blood, then there's potential for a problem such as agglutination, that sort of thing. So in a situation where a female is pregnant, if the baby is RH positive, which is entirely possible depending on the blood type of the father, she can have one child without any reaction. But after she's had the one child that's RH positive, she now has that antibody in her system. Should she have another RH positive child, then it could create problems for both the mother and especially for the baby because they're so small. So due to that, there is a injection given to pregnant women who are RH negative, and I believe that's called Rogam. I'm not sure exactly of the spelling of that, but that's given to females who are RH positive, who are pregnant, and that prevents the possibility of hemolytic disease of the newborn. Okay? So let me pause this again, go back to our other screen. Okay, so picking back where we left off here, the last thing in your outline there says a transfusion, a blood transfusion, is essentially a transfer of whole blood or blood components such as plasma, all right, into the bloodstream. So let's focus now on these white blood cells, or the leukocytes here. All right, here's some pictures of slides, very similar to what you see in Dr. Ward's atlas. So this is from our textbook, helping to identify the different types of slides. So you'll want to know these when we have a online exam for the lab portion of blood, which would be test number 4, I believe. All right, and if we go to this picture back here, all right, we can look at the different structures here. So first of all, we have the leukocytes, the white blood cells. Let's focus on those, all right? They have a nucleus, all right, and they do not contain hemoglobin. So in our outline there, all right, let's start with the granular leukocytes. Now, they're called granular leukocytes because you can see they have granules in the cytoplasm, all right? They tend to have lobed nuclei. They have multiple lobes to them, usually 2 or more lobes, each one, okay? They're referred to, classified according to the type of granules you see in the cytoplasm, based primarily on the color. So the first one we have would be eosinophils, right here. So there's our eosinophils. The eosinophils tend to stain red to orange. The granules, at least red to orange staining granules, usually have anywhere from 2 to 3 lobes. Next up we have basophils. The second one here, remember, are the least common. These have blue to purple granules, which obscure a nucleus that generally has 2 lobes. So we say a bilobed nucleus. And again, see how you cannot see the nucleus here because of all the granules obscuring it there. So the dark blue to purple granules. Next one are the neutrophils, which again were the most common, if you remember that. So these have small pink to lilac color granules, okay? And that's not my name for the colors. That's textbook wise. They refer to those as pink to lilac color granules. with a nucleus that has anywhere from 2 to 5 lobes. So anytime there's more than one lobe to the nucleus, all these lobes are connected by nuclear strands of nuclear material, so they are all kind of hooked to each other. All right. Then number 3, we go to the agranular leukocytes, or the agranulocytes, which essentially don't have granules in them, and we can classify them 2 different ways. First, we have lymphocytes, which you may make a little note here. They can be classified as small or large lymphocytes. And these would include B cells, T cells, and natural killer cells. So slightly how do we designate these? They have a dark, slightly round, slightly indented nucleus. So dark, round, slightly indented nucleus. You can see the indentation right there. Okay, that takes up almost the entire cell. All that's left, we have a thin rim of sky blue nucleus, or sky blue cytoplasm that surrounds the nucleus. Okay, so I kind of messed up that description there for you. So a dark, large, round, slightly indented nucleus with a thin rim of sky blue cytoplasm that surrounds it. And then our last type of white blood cell are monocytes. So monocytes tend to have a horseshoe to kidney-shaped nucleus in it, with what's described as a sky blue to gray foamy appearance to the cytoplasm. And I've never looked at one of these and said, oh, that looks foamy to me, but that's the definition as far as the book goes for those. So functions of these white blood cells. Okay, let's talk about the different functions here. White blood cell functions. Lifespan of these white blood cells are going to range dramatically. It can be anywhere from several hours to years. All right, but we're going to list the average lifespan of a white blood cell as several days. Okay, several days. You don't have to know the number, all right? So the number is not an important thing for you to know in regards to me. But the last part of number 2 there, you need to make sure you know this, okay? The ratio of red blood cells to white blood cells is about 700 to 1. Make sure you know that number. So when we look at a blood smear, right, like we do in lab, you're going to see for every 700 red blood cells, you'll find 1 white blood cell. Okay? Number 3 says white blood cells can leave capillaries by way of immigration. Okay, that means they can actually leave the capillaries and go to site of infections, allergic reactions, and that sort of thing. General function then of white blood cells. Think about when your white blood cell count gets elevated. It's when you're sick, when you have an infection, when you have allergic reactions and things like that. Your white blood cell count goes up. So because of that, it does that because the primary function is to combat microbes and toxins by phagocytosis or some sort of immune reaction, right? Immune response. So let's talk about what each of these actually does, right? Letter A, neutrophils and macrophages, all right? Neutrophils and macrophages, which again are white blood cells, all right? These are attracted to sites of infection by what's called chemotaxis. That's how they get there, all right? And they're active in phagocytosis. So again, you should see, in your mind, you should see Pac-Man running around gobbling up all these different microbes. Basophils, over here, are going to release substances which intensify the inflammatory reaction. And so they're involved with hypersensitivity or allergic reactions. So if you have an allergic reaction to something, your basophil count is going to go way up. Eosinophils are going to help to try to combat that. On the other hand, they're going to release enzymes that essentially combat histamine in allergic reactions. All right? And if you remember from when we studied tissues, histamines that were released from mast cells, all right, or what essentially release histamines, right? Eosinophils are also effective against certain parasitic worms, in case you run into that problem down the road. B cells, which remember the B cells and T cells are lymphocytes. B cells develop into plasma cells and secrete antibodies, destroying bacteria, making their toxins not active. And the T cells, again a form of lymphocytes, attack viruses, different fungi, transplanted cells, cancer cells, some bacteria, but what they're primarily responsible for, they're going to be the ones that get involved with transfusion reactions, allergies, and if you were to get a transplanted organ and your body rejected that, it would be the T cells that are going to be responsible for that. And finally, the natural killer cells attack a wide variety of different microbes and tumor cells that spontaneously show up. Normally when you have a blood test, you're going to go in and you're going to get a white blood cell count, but you won't get a percentage of each one of these individual leukocytes or white blood cells unless you have what's known as a differential white blood cell count. It's more expensive, and when you do that, then you break down and finally and you see what each type here, the percentage of each type of the white blood cells. So it's definitely a more specific test, but it's also a more expensive test and not done on a regular basis. All right, so that's what those look like. So when we get back here, let's look at the platelets really quickly. I don't know if I got a really good picture of the platelets. Here's the blood smear again. So all these you're seeing are red blood cells. Here are the white blood cells, here, 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 here. And all these little fragmented cells between the white blood cells are our platelets. So platelets are what are also called as thrombocytes. A thrombus is a blood clot. Right, so thrombopoietin, essentially what stimulates stem cells to become what's called megakaryocytes, which are very large cells. Those megakaryocytes break up in anywhere from 2,000 to 3,000 fragments of cytoplasm, all right, which end up forming the platelets themselves. You don't have to know the number. On number 3 for the platelets, they are said to be kind of disc-shaped. Our model doesn't look like disc-shaped structure, but they don't have a nucleus either. Primary function here for the platelets is to form blood clots, help to clot our blood. So it says it helps repair slightly damaged blood vessels by forming a platelet plug and promoting blood clotting. Okay. Lifespan. Platelet lifespan is very short, anywhere from 5 to 9 days. Okay. So that should take us through and pretty much cover most of our chapter on blood. You got kind of the basics of that. I have a short video that I will send you a link to. It's an interesting little video on on the different blood types. Okay, so I will try to send that link to you so you can queue up that video. It's about a 5-minute video, but it's pretty interesting to kind of watch that video there. In fact, I possibly could queue it up right here, and let me see if I can do that, and if so, I'll just include it as part of this slide. So let me pause this, and if I can bring it up, I'll just play that, and that'll be the end of this chapter review. Okay, so this won't take up the entire screen, and the quality of it is somewhat lacking, but I'm going to go ahead and turn this on and play this for you as part of our chapter review, because I normally show this during our class time. And I'll send you the link to this if I can, so you can watch it on your own separately if you desire to do so.
Sir, are you all right?
Huh? Oh, hi. Sorry, I'm here to donate some blood.
Oh, all right. Just have a seat over there, and the nurse will be right with you.
Okay, thanks. I've come here to this clinic to donate some blood, but there's something I'm curious about. Have you ever wondered why they use all those letters to describe our blood? I mean, what makes A blood different from O or B? And why is it so important that doctors know what kind of blood we have? Well, it's taken centuries for us to unlock the secrets of our blood, and we've learned some hard lessons along the way. For instance, we once believed that illnesses were caused by bad blood. So to make people well again, we would drain the offending blood from their bodies. As you probably guessed, this procedure wasn't very successful, and for a couple of good reasons. First, here's how much blood is flowing through the average adult body, about 5 liters. Now, if you were to lose a liter or more of your blood, you'd start to suffer from a condition known as shock. When you lose a lot of blood, your body can become starved for oxygen. If that blood isn't quickly replaced, you can fall into a coma and die. And that's what happened to a lot of those early patients who had their blood drained. They died. Now, in one respect, those early blood-draining doctors were correct. A lot of infectious diseases are carried by our blood. But what they didn't know is that our blood also carries the cells we need to fight those diseases. Our blood can be divided into two main components, red cells and plasma. It's the red cells that give our blood its color, but their main function is to carry oxygen to our tissues. Now, if you remove all the red cells from your blood, you're left with a yellow fluid. That's the plasma. And it's here in the plasma that you find the cells that help you fight infections. Now, the discovery that blood was required to fight disease gave the early doctors a new idea. Why not just transfer some blood from a healthy person into the body of a sick person? Well, sometimes this procedure actually helped, but in most cases the result was, well, fatal. What they didn't realize at the time is that not all blood is created equal. You see, a little further along in history, we discovered two special proteins on the surface of our red blood cells. They're called antigens, and we labeled them A and B. Then we discovered that some people's red cells have only the A antigen. Some have only the B antigen. Some have both A and B, and some have neither antigen. Put it all together, and you have the four basic blood groups: A, B, AB, and O. I know what you're saying. So what? Big deal. Well, it's not a big deal. That is, until you try and mix some of those blood types together in someone's body. To show you the problem, let's go back to the clinic. Now, I happen to have type A blood. So let's take a sample of my blood and divide it into two. To the first sample, we'll add a solution containing plasma from type A blood, the same blood type as mine. To the other sample, we'll add a solution containing plasma from type B blood. Now we'll mix everything together, and there's your problem. The first sample looks normal, but in the second sample, the red cells have all clumped together into a sticky mess. So what's happening? Well, floating around in the plasma of everyone's blood are structures called antibodies. Now, the job of the antibody is to patrol your blood, looking for foreign invaders. So suppose I have type A blood, but I get injected with type B blood. When my antibodies see these B antigens, they think this cell is an intruder, so they latch onto it and sound the alarm. And as the intruding red cells and the antibodies collide, they stick together and form a clump. These clumps disrupt the flow of blood to your cells, and without emergency treatment, you can die. Today we prevent this problem by cross-matching a sample of the donor's blood with a sample of the patient's blood. This test ensures that the blood is compatible and that there will be no clumping. However, sometimes in an emergency, we don't have time to cross-match the blood before it has to be transfused. In this case, we have a backup plan. We always transfuse type O blood. Why type O? Well, if you remember, type O blood has neither the A or the B antigen on its surface, which means no matter what type of blood you have, the antibodies in your plasma won't think this is an intruder, and your cells won't clump together.
Okay, so as far as your blood cell type and so let me see if we can go to this. All right, so as far as blood types for our purposes for quiz and testing, we're going to say type O is the universal donor. But at the same time, if you watch TV, you watch emergency room shows all the time, somebody comes in and there's a large blood loss, that person will come in and they'll immediately scream out that they need a certain number of units of O neg, right? O negative blood. So in all really true situations, O negative is the real true universal donor, because no matter if a person is Rh positive or Rh negative, they would be able to receive O negative blood, right? If you have a situation where somebody's Rh positive, in other words, they had like A positive or B positive or AB positive blood, were they A negative, B negative, or AB negative blood, and they were to receive positive blood, all right, then they could possibly have that situation with, as in hemolytic disease, their blood could agglutinate. So that's why O negative is the real, true universal donor. But once again, for our test purposes, we'll just say type O blood is the universal donor. All right, so that takes care of this chapter, and we'll move on from here. And the next thing you'll see as we go to will be heart. All right.